Good afternoon, my dear kids. Today I have taken a topic from class 12th, that is chapter number 6, molecular basis of inheritance. In the previous chapter, we have seen how Mendel has given the pattern of inheritance, but he was not able to make out what factors were responsible because there were restrictions in that era and not that much of technology was not there. They were not able to elucidate the DNA molecule, which is now considered as the genetic material for all the living organisms. So now in this chapter, molecular basis of inheritance, we'll be looking inside the molecule, which is the base of the inheritance. Okay, so we know in the previous chapter, we have seen in the school in the last lectures, we have studied about how DNA was being said as the genetic material. Okay, so now we'll be looking inside the DNA molecule, what DNA molecule is. And in class 11th, you have studied about DNA molecule is made up of nucleic acids, okay, and there are two types of nucleic acids, the DNA and the RNA. The full form of DNA is deoxyribonucleic acid, and it is obviously a acidic substance. It was first identified in the year 1869 by scientist Friedrich Mister and named it as nuclein. And at that era in 1869, there were less technical advancement which poses restrictions in elucidating the long DNA polymer molecule. So now we have seen in our previous lectures the transforming principle being conducted by Griffith in 1928, taking up the inference that DNA is the genetic material. So now we'll be starting with DNA is a polymer of deoxyribonucleotide. Polymer means, mer means unit, poly means many. So that means it is a repeating unit of many nucleotides. So first of all, students, you should know what do you mean by the term nucleotides. So nucleotide is what? Nucleotide is composed of three components that will be taking up in the next lecture. So first of all, we should know what is the length of DNA. The length of DNA is the number of base pairs. We'll be learning about the Chargaff rule in the next lecture. Then we'll be telling about what do we mean by the number of base pair. It is the complementary ratio between the adenine and the thymine, between the cytosine and the guanine. These are the some nitrogenous base pairs. So as we take about some example, the length of the DNA is the characteristic of an organism. Okay, so example we are taking as bacteriophage. It is also known as 5 into 174 and it has got 5386 nucleotide. So that means this number will always be constant. Another example we are taking it as bacteriophage lambda. It is having 48502 base pairs. So all these bacteriophage, what are bacteriophage? Bacteriophage are the viruses which infect the bacteria or which eat the bacteria. Okay, bacteria, phage. Phage means to eat. Next example we have got Escherichia coli. It has got 4.6 into 10 to the power 6 base pair. So that means as compared to the bacteriophage, E. coli will be having more number of DNA molecule. Another example we are taking is of haploid human DNA. It has got 3.3 into 10 to the power 9 base pair. Haploid means single set of chromosomes in the human DNA, and these are given in your NCERT textbook, and these are very important as they can be asked in 1 mark question or they can be asked in the MCQs. Now in the next lecture we will be moving forward and we will be learning about the double helix of the DNA structure and structure of the DNA polynucleotide.